In 1975, scientists succeeded to generate monoclonal antibodies for the first time, leading to a scientific evolution in the medicinal field. Nowadays, monoclonal antibodies are one of the fastest growing biotechnological derived molecules that are currently used in the treatment for many several diseases. But what are the monoclonal antibodies? In contrast to the polyclonal antibodies, the monoclonal antibodies are monospecific, made by unique and identical immune cells. They are characterized by being very specific, or by another way, they have monovalent affinity to a specific binding site on an antigen, which is called the epitope. The first method for production of monoclonal antibodies took place on rats by a technique called hybridoma technique. The hybridoma technique involved the introduction of certain antigen binding plates to the model. After that, the beta lymphocytes are obtained from the model spleen. Fusion of the beta lymphocytes with an important cell lining, like any immunoglobulin producing cells, then took place, for example, the myeloma cells. The next step is culturing the hybridoma in a specific medium like the HAT medium that is selectively growing the indeed hybridoma. The primarily produced hybridoma contains several antibodies produced from different clones of beta lymphocytes, each producing its own antibody. Dilution then can take place in order to separate different clones in several wells, which are screened to reach the needed antibody. Then cloning and testing takes place. As for the advantages of the monoclonal antibodies, firstly, I'm going to talk about the homogeneity of the monoclonal antibodies. The monoclonal antibodies represents a single antibody molecule that binds to the antigens with the same affinity and promote the same effector functions. Their specificity, the product of a single hybridoma reacts with the same epitope on antigens. Their selection, it is possible to select for specific epitope and generate antibodies against a wider range of antigenic determinants. Antibody production, unlimited quantities of a single well-defined monospecific reagent. Also, another advantages for the monoclonal antibodies is that it could be used in the treatment of various number of diseases rather than cancer. The monoclonal antibodies examples are divided into two parts, the new convention and the old convention. The new conventional examples starting with olaratumab, which is an anti-neoplastic. Its name is composed of the components olara, T, U, mab, which shows that the drug is humanized monoclonal antibody acting against tumors. Second one, the name of benaralizumab, a drug designed for the treatment of asthma, has the component of benaralizumab, marking it has a humanized antibody acting on the immune system. As for the old convention examples, starting with the adalimumab, which is a drug targeting the TNF-alpha, its name can be broken down to adalimumab. Therefore, the drug is a human monoclonal antibody targeting the immune system. If adalimumab had been named after 2009, it would have been adalimumab. The second one is abciximab, which is a commonly used medication to prevent platelets from clumping together. Broken down to abciximab, its name shows that the drug to be a chimeric monoclonal antibody used in the cardiovascular system. This and the following two names would look the same if the new convention were applied. The third one, the name of the breast cancer medication is trastuzumab, can be analyzed as trastuzumab. Therefore, the drug is humanized monoclonal antibody that is used against tumors. The last one, alasizumab pegol, which is a PE glycated humanized antibody targeting the circulatory system. The applications of the monoclonal antibodies are classified into three types. First, the diagnostic applications. The monoclonal antibodies have been successfully used in laboratory diagnosis of several diseases, and so they are used as diagnostic reagents in analysis or as tools for imaging diseases. Some tests that are based on the monoclonal antibodies are used in radioimmunoassay and ELISA in laboratories. those who measure content of some hormones like insulin or growth hormone, and other tissues or cell products like the tumor markers or the blood group antigens. And so they are now possible in early diagnosis of pregnancy, cancers, hormonal disorders, and some infectious diseases like antigens of Neisseria gonorrhea. The second application is the therapeutic applications, which can be classified into monoclonal antibodies as therapeutic agents and monoclonal antibodies as targeting agents. To start with, the monoclonal antibodies as therapeutic agents, which can be used for enhancing the function of the immune system of the host. Moreover, direct use of monoclonal antibodies cause less toxicity to target tissues or the host. For example, destroying the disease, treatment of cancer, treatment of AIDS, and immunosuppression in organ transplantations. The second one, the monoclonal antibodies as targeting agents. The monoclonal antibodies conjugate with toxins, drugs, or isotopes and carry them to the target tissue for the efficient action that allows higher concentration of the drug to reach the desired site with minimal toxicity. For example, monoclonal antibodies as immunotoxins in drug delivery and in radioimmunotherapy. The third application is the protein purification. Monoclonal antibodies can be produced for any protein and so can be used for purification of protein against which it was raised. This can be prepared by coupling them to cyanobromide activated saccharose. The immobilized monoclonal antibodies are very useful for the purification.